All right, hello everybody. I'd like to welcome you to Pediatric Grand Rounds. I'm delighted today to have with us as our speaker, Dr. Atul Boot. Dr. Boot trained in computer science at Brown University and also worked as a software engineer at various places, including Apple and Microsoft. He received his medical degree at Brown University and then made the great decision to become a pediatrician, and did both his pediatric residency and his pediatric endocrinology fellowship at Children's Hospital in Boston. And then he went on to get a PhD from Harvard and MIT. Dr. Boot serves as the chief data scientist for the entire University of California health system. And he's built a really amazing research platform around that I imagine is going to tell us more about today. He's been continually funded by the NIH for 20 years. He has 24 patents to his name, and he's authored over 200 publications. He's been featured in major lay newspapers such as the New York Times, the Wall Street Journal, Wired Magazine. He was elected to the National Academy of Medicine, and he was recognized by the Obama administration as a White House champion of change in open science for promoting science through publicly available data. He's also founded at least 3 companies, perhaps more by this point. And so it's with great excitement that I welcome you, Atul, to give Grand Rounds today.
Great. Well, Rafi, thanks for having me. And I'm going to start with an apology because I've been in the department for more than 5 years, and I'm finally giving Grand Rounds. So probably long overdue. And I was also noting, amusingly, that we set up this date a year ago. So who knew where we were going to be a year from now, or back then? So I thought we would be in person probably when I said yes to this a year ago. So anyway, long overdue, and virtual is better than nothing, I guess, is the way to start with the punchline. All right, so I'm obviously a medical doctor. This is a medical audience. I got to start with my conflicts of interest. I have just a few. Okay, perhaps more than a few. Suffice it to say, I've started a bunch of companies. I consult for a bunch of companies. You might not want to believe another word I say over the next 50 minutes. I wouldn't blame you. But I'm really proud of the right side of the slide here. Those are all the companies started by my students. Now, they don't always include me in their companies, nor should they. But in general, I'm really proud of that because in my lab, we probably have a couple sayings. And one is that if you want to change the world, you can't just keep writing papers about it. At a certain point, you're going to invent things or discover things. And if you really want to change the world, you got to deliver that to patients, to families, to doctors. So I take pride in the fact that we invent these things, and then sometimes they're useful when you actually spin them out of academia into companies, let's say. And I'll talk about that. And it's not taboo to talk about companies. And it's probably even important to talk about them. So a lot of doctors use a slide like this as a slide of shame. This is my slide of significance. This is the impact that we're going to have in the real world, I think. But anyway, with all that, let's get started. If you haven't noticed, you must have noticed that we're in this big data era. You pick up any of these major magazines, you see big data and AI and machine learning. Of course, it's an incredible time to be thinking about all the data we're collecting. Some of this seems scary. Some of this seems incredibly potent, and there's huge potential. I note with some humor that The Economist magazine covers this again and again. You can see at the bottom row there. The bottom right one was the most recent, where they're equating data to oil, right? These are oil rigs there, and they're saying the data is the most valuable resource. Now, people say this, data is the next oil. I hate this phrase because oil to me is very grabby. Either I get the barrel of oil or you get it. We're not both going to have the barrel of oil. But data isn't like that. I might get data and do something with it, and you might get the same data and do something completely different with it. So it's not like oil, right? You can divide this without losing what's in data. So instead, I like the saying that data is the next soil, that you use that soil to plant your ideas and then watch them grow. Probably a little bit more holistic there. But anyway, let's just level set what all these terms mean, and especially AI and machine learning. Make sure we learn something in this Grand Rounds. These are the three phrases that are bandied about a lot. And this is my framework, one way to framework these phrases. To me, artificial intelligence is the broadest of these phrases, mimicking aspects of human intelligence with computers. So it's computers driving cars by themselves, computers translating languages, right? That we've seen all of those examples recently and over the past decades. And then machine learning is one kind of artificial intelligence. And machine learning in particular is, let's get the computer to do AI by giving it data, by teaching it with data. Now you might think about that and say, of course you're going to use data. How else would you teach a computer? Well, in the old days, meaning 30, 20 years ago, we didn't have a lot of data to give to computers. So we would make rules for the computers to learn, right? So you would ask experts, even doctors, well, what do you do when you see this? And what do you do when you see this? And we would dutifully write down all those rules and then teach the computer the rules. And I think what people have now figured out finally is that it is much better, for example, in the medical world, to show what a doctor is doing with the data instead of asking the doctor, what do you do in this case or that case, right? The data is the actual evidence of what's happening in the field. So we used to be rules-based, now we're very data-driven. Now, under machine learning, you have supervised and unsupervised. And supervised means that there is a right answer, right? So the radiologist has a right answer what that chest x-ray looks like. The pathologist has a right answer. And so we can score the computer into really telling, is it close enough or far enough from that, right? Accuracy, sensitivity, specificity, ROC curves, precision recall, the computer scientists use a little bit more. So we're used to supervised questions. We know how the humans do as a gold standard. How can the computer do? Unsupervised means we don't have a right answer. We're drowning in a lot of data, drowning a lot of samples, and let's ask the computer, what structure does the computer see in there, right? Clustering, k-means clustering, things like that, where there's no right or wrong here. There's no sensitivity or specificity. Just come up with some way to organize it. That's unsupervised. And then one kind of machine learning is deep learning, where crudely the computer software code looks like layers of a brain, right? With one input going, one output becoming the input for the next layer. It's a very crude analogy, but that's what it looks like. So it's one kind of deep learning. It's in particular great at learning lots of non-linear types of connections, especially images, sounds. You've seen the deep fakes that are going out, the videos now. It's incredible with enough computational horsepower and memory. The deep learning methods are really gaining in popularity and ability. Now, this all seems mystical. Let me demystify all of this. All of these are software tools that you often borrow from the internet and put in your software tools, right, that you're building. So, or in other words, there are dummies books literally for all three of these fields, okay? $17 each on Amazon Prime, right? So there is no wizard behind the curtain here. It's just software tools that we commonly use and then put into our own software to make these seemingly magical things happen, right? But it's not, there's no magic here, it's just software. Now, of course, it's changing medicine already, right? So whether you believe there's 3 ways or 300 ways it's already changed medicine, on the right there, you have Eric Topol's book, Deep Medicine. What I thought were seemingly very molecular journals like Nature Medicine are now about data and machine learning. JAMA has calls for proposals. We have very old journals like Artificial Intelligence Medicine now all of a sudden are popular again. And then you had fields like even the field of radiology has created a new radiology AI journal. So you see that happening a lot. I've not yet seen pediatrics AI yet, but one could imagine that could happen. And of course, it's not just the academic world, it's the industry world as well. Here are the first 3 big tools that were approved by the FDA on machine learning and AI, specifically on deep learning. And you can see the earliest at the top there is just January 2017. That was not long ago, 3.5 years ago. These are the first 3. So the one in the back there is a company called Arteris that looks at cardiac imaging, trying to figure out cardiac status. And actually there's a Stanford and a UC connection for that one. Viz AI was for stroke triage. You know, does this patient need emergency therapy quickly? And the diabetic retinopathy is the third one. And you can imagine all the retinal images people get, not enough ophthalmologists, maybe software can make that call. And many people working on diabetic retinopathy, including Google itself. Now, these are the first 3. Here are more than 40 that have been since approved, right? 2018, 2019. Already this slide is 1.5 years old. There's a link there that takes you to the article about this. And you can see on the right there, all sorts of areas of medicine. So it's not just radiology, many areas being approved constantly now. So this is an incredible time. The FDA has told the field how AI and data are all going to be used. This is the time to really do as much as we can to create tools and techniques here. Now, of course, this seems new and hot, but it actually goes way back. So these are some of the earliest papers on AI in medicine. Ted Shortliff is a founder of our field of medical informatics. And you can see in the early 70s in computers and biomedicine, it was so new back then, they literally put the software code into the paper, right? It seems so magical back then. Antimicrobial therapy, look at resistance patterns. You give in the input and the software would tell which antibiotic to use. We carry those pocket cards around, probably still, maybe it's all software now at this point. But it's funny on the right there, medicine and the computer, the promise and problems of change. That could be a title today in the New England Journal of Medicine. This is from the early 70s. And what happened is, of course, there was AI hype in the 70s, and then all came crashing down. The expectations just couldn't be met. There was just no way for the field to really meet all the promises that were being promised back then. But it is all back now. And why is AI back? Why are we talking about all these things constantly, even at UCSF? These are 4 big reasons. First, we have incredible hardware from the video gamers. Yes, of course, people love playing video games. And the point of video games is to get those bits and all those graphic elements onto the screen as fast as you can. Well, it turns out that same kind of hardware, those same kind of chips that are great for working on video like that, are excellent for deep learning as well. In fact, so much so we buy so many of these chips, but sometimes gamers have a hard time buying their chips or buying their boards. But companies like NVIDIA, just south here in Santa Clara and San Jose, are just cranking out those chips like crazy. I mean, the market capitalization is incredible because you got the deep learners and the gamers using the same kind of hardware. But it's consumer pricing for hardware. It's incredible. Then you got software libraries, which are free from companies like Google, which know how to write software, and then Berkeley and others really coming up with excellent software packages that are given away for free. So incredible communities have sprung around the software packages. You even have high school students now. This is a common high school summer project. Is to use machine learning to do this kind of, to do interesting projects like that. And then in medicine in particular, we have 2 enormous data sets now to train computers with. Now I'm going to talk about both of these in particular. One is the molecular field, molecular data, molecular genomics, DNA, other molecules. And it's not just measuring more things, we're measuring them on more people, right? So you got the molecular world in medicine, and you got the electronic health records side, where we're literally writing down into a computer everything we're doing and measuring from a patient. And I'm going to spend time on both of these in particular here. And then, of course, we still have many hard unsolved problems. Patient predictions, what's going to happen next for this patient? How do we get to efficient drugs and drug discovery? And for this population, let's say have patients with asthma or diabetes, what's the right best way, the best, safest, cost-effective way to treat this population? These are hard problems. All of them still need solutions here. Let's see here. All right, let's get into the molecular world, because this is how I got my start way back as a peds endocrine fellow in the late 90s. I had to start with my research here back in Boston Children's, and these are still my favorite way to talk about big data in the molecular world, these gene chips. This is how I got my start in Zach Kohane's lab in 1998. These gene chips, this is my hand, I think, holding one of these, are just one of many examples of how we measure molecules like crazy from patients. Right now, I'm showing you these gene chips. We happen to measure RNA levels, but of course we measure and sequence DNA. I could have shown you blinking lights from a sequencer. I could show you genotypes. I could show you protein measurements, mass spectrometry. I love this one because this is how I got my start. And you could see immediately we got bored of measuring the 1 by 1. People started to buy the 96-well format of these chips, right? So these technologies just keep scaling every couple of years. And I love this technology because for a couple $100, you can put a piece of cancer on there. Of course, you got to process that, and you get a readout of every gene in the genome, which genes are up and which genes are down. Incredible time. Now, what happens a lot in the molecular world is that we get these nifty technologies. And what happened in this world is everyone started writing papers, right? When I was a fellow and then a junior faculty at Boston Children's, everyone started to look at how to analyze this data, right? We were on dozens of grants for this. And then we started to write papers and submitting papers. And you know what happened? The journals started to get overwhelmed with these papers, right? Here's the list of genes that change in asthma. Here's a list of genes that change in cancer. Here's a list of genes that change in heart failure. And then the journal said, you know what? We're getting way too many of these papers. No more papers with gene chips. That's it. Okay, that's it. Unless you put all this data in a public repository, right? Because the reviewers have got to double check all the math now. And maybe others can use these samples for something else. And so that's what happened in this field. You know, the precedent for this goes back to something called GenBank, right? I know you all use PubMed. GenBank is just a next door neighbor to that, right? On those tabs at the top. Well, GenBank is now, what, 40 plus years old? Because way back when the first DNA sequences were shared, nobody wanted to type AGTCs all over again. So they were sending tapes to each other. But of course, what it means is if enough scientists use a molecular measurement, eventually all that data is got to get to be publicly available. And so that's what happened in my field here. Starting in 2000, 2001, 2002, the NCBI, which is the National Library of Medicine, they run PubMed too. They built a repository called GEO. The Europeans built one called Array Express. And then fast forward to August of 2012, when we hit 1 million samples publicly available, right? So I started my postdoc and fellowship in 2000, even before that, and boom, you can see in 12 years, 1 million public samples. These are samples of cancer, mouse models, cell lines, human, mouse, all sorts of species, publicly available. Now, if you look now, I think it's already past 3 million. So just skyrocketing up, right? These are all de-identified, but these are all samples waiting for you, okay? Because what most people do when they get a big grant to go measure these molecules, right? They go measure a bunch of molecules. They go write a couple papers. And then what do? They go get another grant to go measure more molecules. And meanwhile, all of this data eventually has to be public. And all of it is sitting, waiting for you. If you learn how to use this data, you got more samples and more data than any researcher contributing data in here. So let me give you one example of such a repository. This is one that I know and love called the Cancer Genome Atlas, okay, TCGA. This is a more recent slide than I'm used to, but here you can see nearly 85,000 cases of cancer of 67, 68 different kinds, okay? This is where the data is coming from, and 22 types of data. So you got molecules, of course, like DNA, oncogenes, you got RNA measurements, protein measurements, you even got clinical data, which medications, which chemotherapeutic agents, which targeted therapeutics were they treated with. Of course, you got sequencing data. In some cases, you have mammography for the breast cancer. You have brain scans for the brain cancers. And all of this data, more or less publicly available, waiting for you. And meanwhile, TCGA keeps growing. Now, actually, there are UCSF cases in here. I think ovarian cancer, prostate cancer, the TCGA came from UCSF, right? So some of our patient data, de-identified, of course, is available through these repositories. And so this is just 84,000 cases here. Can you imagine millions and millions of all these other disorders already there too? I'm going to put in a plug for one repository that I know and love because I run this one called Import. To me, the next big open data is going to be clinical trials data, right? We all know and love clinical trials. We run a bunch at UCSF and across the University of California. And I think that raw data is going to be publicly available too. So we have more than 300 trials here. What are we going to do with clinical trials data? We're still all learning. Can we figure out, why does the pharmaceutical company think this drug is working or not working? Maybe patient subsets for precision medicine, which we'll talk about in a moment. Comparing one drug against each other. I think there's going to be a lot of future here for clinical trials data. So sign up and Import, and you too can start to download and play with clinical trials data as well. So that's this new role and new realm of open data. What open data is going to mean for all of you is this data is available for you to launch a research project with, right? You don't have to go start with an IRB. You don't have to start with recruiting patients. Maybe someone's already made measurements that are relevant to you, and you can discover something in this data that they've completely not seen or completely missed. And all of this is really heading towards precision medicine, right? So here's President Obama mentioning precision medicine. And look, it's been almost 10 years now since the seminal report from the National Academy of Medicine even coming up with this phrase, precision medicine. It's a great report. It's still very timely. Of course, Keith Yamamoto and a whole bunch of precision medicine platform folks work every month to keep us on the edge, the bleeding edge at UCSF in precision medicine. But if you actually distill this entire report, what it comes down to is really to reach precision medicine, we're going to need three big circles at the top here on this slide. We're going to need better diagnostics. We're going to need more and better therapeutics. And we're going to need to figure out health outcomes. So the entire rest of this talk is basically on these three circles. How are we going to come up with better diagnostics? How are we going to come up with better treatments? And boy, how are we going to even figure out all the health outcomes here, okay? And I'm just going to step through what my lab has been doing, what I think we should be doing to use data, not just molecular, clinical data, to get to these three circles here. All right, that's the rest of the entire talk here. And of course, going to save time for questions here. All right, so let's get to diagnostics first. And so what is a diagnostic, right? So a diagnostic is a way for clinicians, for health providers to really tell if a patient has a disease or condition, right? We have imaging diagnostics, right? I love blood tests, right? Because they're a little bit, seem to be more specific or more sensitive. And way back in my Stanford days, now more than 5 years ago, we wanted to come up with a new blood test for preeclampsia. Okay, let's pick a pediatric disorder here. And to me, preeclampsia is still one of these conditions that falls between the cracks, right? It falls between the cracks between OB and pediatrics, right? Because I love the obstetricians, right? They're great. I have good friends who are obstetricians. But it also used to annoy me that they would say, the delivery is the cure. Of course, from their perspective, the delivery is the cure. But then we pediatricians have to deal with these tiny babies, make sure their brains stay healthy, make sure they stay free of infection, right? And all of this leads to incredible mortality and morbidity across the world, across socioeconomic groups, across cultures and races and ethnicities. You can see some of the costs estimated in one way or the other here. So, and what is our test for preeclampsia, right? There are a lot of groups still working on this, but in general, it's just urine protein, right? It seems like the most non-specific test we have in medicine. So we wanted to come up with a blood test for preeclampsia. So Linda Liu was a grad student involved with this. Bruce Ling is still a good friend and collaborator at Stanford, and Matt Cooper got involved, as you'll see in a moment here. And so what do? Is we start by just literally typing in preeclampsia and preterm birth into some of those databases I showed you already in the talk. And we find dozens of the data sets already available. And they're in the RNA world. So maybe the RNA is coding for proteins. So let's just put all that data together in a big Venn diagram. I forgot what's in common here, right? I don't believe it if one researcher sees it. I want 7 researchers. I want 10 researchers to see the same markers going up again and again and again. And then let's go build a test for that, right? Let's go make a protein test, what's called an ELISA, okay? It's like 1970s technology. And so here's kind of the punchline of one of the papers here. First, you can see it was funded by March of Dimes and what we call SPARC at Stanford, what we call Catalyst at UCSF from the CTSI. Here's one example of a marker called hemopexin, and it's up in the pink, which is preeclampsia compared to normal, healthy pregnant women, which is in the green. You can see some p-values there. And all of these were papers that you can see a link at the bottom right there. So look, we got some grants, we wrote some papers, and what do you do next in Silicon Valley? You start a company on this. And this became a company called Carmenta. So this was started within Stanford. And then we got $2 million of seed financing, as you can see at the bottom left there. Now, just to be crystal clear for a moment, $2 million isn't a lot to raise in Silicon Valley. But it's enough to launch a prospective clinical trial, right? And that's going to be one of the punchlines here. What's the next scientific study that needs to happen was a prospective multi-center trial. But that science is now going to be completed and conducted by a startup company, not in my lab, okay? Or to put it another way, do you know how hard it is to get a $2 million NIH grant today? Sometimes the science is easier to run in the startup company instead of our own labs. And it's okay to do it that way. In fact, our universities are fine with it and want us to do it. Of course, there are a bazillion rules. Actually, there are just 6 rules you got to follow. But that's the point, is at a certain point, you've maxed out what you can do academically. You're going to have to start the company, or someone's going to have to license this in to take it to the next level. Now, I'm really happy to note at the bottom right, this company already got acquired. So we went from starting the science, downloading the data, getting the markers, doing the informatics, testing the samples in the freezer, with Eliza's, launching the company, selling the company, start to finish, 18 months. Of course, the inventors are happy. The investors are happy. Of course, we're going to do more of these. And I'm giving away the secrets here, guys, because any one of you in this audience could take a different disease. And man, we would never step on each other's toes. You know, we need that many diagnostics still in medicine. Everything I just said and showed you is in the links here, okay? Step by step, all of the papers, how did we actually get the company launched? It's all in the links here, okay? Trying to show it can be done, and I think we need more people to do this. Let's talk about another diagnostic, DNA sequencing. Oh my God, it's a no-brainer. We're gonna be sequencing more DNA, right? Of course, the human genome was completed in 13 years. It took $3 billion in factory-like settings, like the bottom left at the Broad Institute in Cambridge, Massachusetts. And on the right here is my hand holding an Oxford nanopore. Literally, the sequencer is powered with a USB cable, right? You can look at the difference in 10 years. It's a no-brainer. We're going to be sequencing more patients in medicine. It's just a no-brainer. Now, I'm not saying we're going to have a USB stick sequencer in every clinic room or even in every clinic, but you know, we're probably going to be sequencing in more and more ways. It might not even just be germline. It could be for infectious disease. We'll talk about cancer and all of that in a moment. And so I saw this happening. I saw this coming way back in 2008, 2009. And I got my start because of this particular weird patient named Steve Quake. Now, he's a very unusual patient. He happens to be a bioengineering faculty member, and he starts sequencing companies on the side. And so he was one of the first faculty members in the late 2000s. There was the first this and the first that. I think he was the first faculty member to have his whole genome sequenced. But then he became a patient. And on the right there, you can see that his family tree, he's in the circle, the red circle here. And what happened is one morning, his nephew, age 19, died, sudden death. Okay, so you can see where the red circle is, that's Steve, and the nephew is in the blue circle. And so Steve went to his friend, our friend, cardiologist, Ewan Ashley, and said, well, doctor, am I gonna die of sudden death? And by the way, doctor, here's my whole genome sequence. Now, can you imagine if someone still brought a genome sequence to your clinic today? Like, what would you even do? So this paper in Lancet, it came out in 2010, became a prototype of what's it going to be like for doctors to deal with patients who have their genome sequence. I'm not saying we got everything right, but boy, we had an incredible team-based approach to really come up with a prototype of what it was going to be like. I think this ended up being one of the most highest cited papers in all of 2010, was this paper on patient. This was the first patient to show up with the whole genome sequence. Now look, what do we do? Well, and by the way, all of this has been publicly reported by Steve and others, so there's no HIPAA thing here at all. So look, we got to deal with the heart first. Well, it turns out it's great. Steve does not have any mutation in any known gene that codes for a protein that leads to arrhythmias, let's say, or sudden death. Now, Steve does have a variant in a gene that might set him up to have a myocardial infarction or coronary artery disease later in life, and many in his family have had that, right? So that's different though, but at least he doesn't have any mutation that leads to an arrhythmia younger in life, okay? That's fine. So the heart was done. Russ Altman could give an hour-long talk on pharmacogenomics. Now that we have your genome, what else can we say about it? Well, 150 drugs we can say something about. Would the drug work? Could there be side effects? What dose would we use, for example, for blood thinners and warfarin and all the rest? An hour-long talk that could be there, the 150 drugs we listed that we could actually say something about. So the heart was done, and the drugs were done. And then my lab was stuck with all the rest of medicine, every other disease. Now, it turns out we knew this was coming. And like I said, in 2008, 2009, I knew someday a patient was going to show up with their genome. And so what we did in 2008 in the summers, we literally started with the San Jose high school kid. And we said, okay, here are some papers, right? Because what we realized is there was no master database of just every SNP, every polymorphism, and every disease. There's just no master list. So we gave him 50 papers to read, and you know how smart high school kids can be in the Bay Area. He read all 50, and we started to build a data model. And the term is called curation. You start to curate all these papers. And then we realized, okay, we need to write down these numbers. We can ignore those numbers, but we got to get to race and ethnicity. Where exactly was the study done? What are the p-values? What are the confidence intervals? What is the exact SNP? We don't just want the gene. We want the exact mutation or SNP in there. So we started with 50 papers with him, and then we scaled. We said, well, how much and how long would it take to just read every single paper in human genetics? And that's what we did. And so here are 1,000 papers. I got this way to get my PDFs to look like this. This is 1,000. We blew past 20,000 papers we manually read in human genetics to make the master list, okay? I still argue we probably have the best master list of every gene in the genome, every SNP, and every single disease. And with that, then we were able to compute for Steve Quake, what were his risks of disease going to be like? And this is what it looked like in the paper. And you have font size, right? The bigger the font size, the more likely of the disease, and the smaller the font size, the less likely. And by the way, you know, some diseases lead to other diseases, right? So just because hypertension is small there, he's already set for obesity and type 2 diabetes. If either of those two happen, the hypertension might happen as well. And all along the edge there, we parsed the heck out of PubMed to figure out what environmental factors or behaviors should a doctor recommend to try to alter the etiology of those diseases, right? So of course it's going to say smoking, exercise, diet, and alcohol. You don't need a genome sequence to know those 4 big ones, but all the way tiny at the top, there's pesticides, because he's got a tinier risk for Parkinson's disease, and there were 10 papers linking pesticides to Parkinson's. So we said, well, Steve, you know, if you like to garden, stay away from the pesticides here, right? And so this is our prototype of how a doctor and a patient are going to deal with their genome, right? Not because of AGTC and this genome spot, but maybe it's behaviors and risk factors that can be altered to try to prevent those diseases. Of course, we're using genome sequencing in many other areas now. Neurology, unexplained disorders. You got Stephen Kingsworth sequencing kids in the NICU over at Rady Children's. We're using it in cancer therapies, late stage and even earlier stage cancers. But we have all of this work, and we had a whole bunch of people, faculty members involved. And what do you do in Silicon Valley? We started a company called Personalis, right? So you can see the arrow at the top. We got all the papers curated. 3 other faculty joined with me, and then we brought in the CEO. So all 5 of us, 4 faculty, 1 CEO, were the founders. We ended up raising $76 million until our IPO last summer, which brought in another $141 million. And today, this is a little bit old, the company's worth $1.1 billion. Or again, to put it a different way, the science continues in the companies. Do you know how hard it would be to get a $1.1 billion NIH grant? There's nothing wrong with saying, I can't take this any further in my lab. I don't want this to die. I want patients to benefit. I want doctors to benefit. You start the company to keep the science going and growing. That's it. You gotta follow all the rules. I'm not saying not to do that, but their rules are there. And you know, by the way, the rules are the same everywhere. Stanford, Harvard, UC, our rules aren't any better or worse. Otherwise, I would have never come here. Okay, I read every word of every rule here. Okay, so we'll talk about that when we get to questions here. All right, so that's diagnostics. What about therapeutics? Oh my God, what a mess. It's costing a fortune to make a new drug. And now everyone thinks it takes 10 years and $1 billion to make a new drug, right? We've heard of all that, right? Boy, it costs a lot more than that. Matthew Herper does this in Forbes every couple of years. It's probably a couple of years out. It's probably worse than this now. And when he tallied it up, right, it's really simple math. How much did you spend? Divide by how many drugs did you get, right? Simple math. If you do that math, you can see these top 12 companies spend between 3.6 and nearly $12 billion per drug. Probably not sustainable, right? How are we going to sustain that? So we got to come up with better ways to come up with drugs. Now, I'm not saying we got the magical solution here, right? But can't we use all that data I just showed you on all these samples and diseases and enormous amount of drug data publicly available? And that's what we started to do, is put all this drug data and disease data together. And where we start to have a lot of traction isn't just coming up with new drugs for diseases, but can we find new uses for old drugs that might actually be able to be used for treating diseases? Now, we end up having more than a dozen papers, and they're still coming out in this field. Here's one that's probably one of the more recent ones, and it gives you a great example. And this is Bin Chen who did this work. He's now a faculty member at Michigan State. A lot of collaborators here. Here's a disease called hepatocellular carcinoma, right? Liver cancer. Now, liver cancer affects 350,000 people a year, but Most of them are in India and China, right? This is viral etiology. This is maybe alcohol and other toxins. Not a lot of people in the United States get liver cancer. Now, I don't know if that's a cause or effect here, but I think because of that, we barely have any companies working on liver cancer, right? It's just not a cancer that's really marketable here. But the world needs a drug for this. So turning all of our cranks, we realized that the drug nicolosamide might make a difference here. Now, nicolosamide is used for worms. It's actually used in pets for worms too, right? Worms, I mean GI tract worms. So here you can see an example of a mouse liver treated with human liver cancer samples or liver cancer cell line. On the left is the control. It looks kind of ugly. Nicolosamide actually doesn't look any better, but NEN is nicolosamide ethylolamide, particular salt of nicolosamide that you could take orally, or rather the mouse takes orally. Mouse is eating this with the chow. You can see the cancer scenes better. Here are glowing cancer pictures before and after treatment. Seems some of them might seem to be getting better with nicolosamide. Now, it turns out there is already a drug for liver cancer. It's not a great drug. It's a drug called sorafenib. Actually, it's a UCSF drug out of Onyx, which I think got acquired now. And so here you can see a control, so a mouse treated with nothing. Nicolosamide is the second line. Sorafenib is the third line. And then nicolosamide sorafenib actually together does better than either one separately. So that kind of means there might be synergy here. And so what that means is if I were to run a clinical trial, let's say in China or India, maybe we should do a combination trial here to see if they're actually going to do any better here. Now, this is one of, I don't know, 15, 20 papers we've published like this, right? And in fact, we're able to launch one clinical trial for a different disorder, psoriasis, I think, for lung cancer. And we've got many more that we want to do. And this field is completely exploded now, data-driven drug discovery here. What did we do? In Silicon Valley, we started a company on this called Numedi. This has raised more than $10 million. And they're working for a nasty condition called idiopathic pulmonary fibrosis. 2 drugs there, neither of them work really well. The field needs more drugs. We're really trying to show how this data can come up with a new drug and try to launch it into clinical trials here, right? The science continues in the startup company. Yes, you're gonna keep hearing me say that again and again. All right, so let's switch gears a little bit out of that and think about clinical sides of things, right? And so let me just reintroduce what I've been building here for the last 5 years here. I'm really privileged to be able to take over the leadership for this Baker Computational Sciences, Baker Computational Health Sciences Institute. Our mission really is to build the strongest team in the world at UCSF in biomedical computation, health data intelligence. We're the academic home for this kind of faculty member. They usually know who they are. They might not be in a computer science department here, it doesn't matter. 70 faculty across all 4 schools. We're recruiting like crazy. I'll show you the graphic in a moment. We're building educational programs, which I'll talk about. And all this is really to help research and development continue and use these data assets in a stewarded, principled way here. In keeping with the recent Twitter theme of how it started, we were in Mission Hall, literally Pede space, right, on the 4th floor. I know most of you don't go back to Mission Hall right now, but we've left now. We've moved right across the street, kitty corner, to the new, what used to be called Block 33, or the Valley Center, the Valley Tower. So when we're past COVID, you'll be able to just enter in from the north, but right now you got to go all the way around the building to the south entrance because that's where the guard is. And of course, now we finally have labeling, so we're actually on the map there with the signage outside. If you come to the 2nd floor, this is literally what it looks like, and we're the entire 2nd floor of the Valley Tower on that side. We're working on talent, training, and tools. And by the way, with Venn diagrams, I know there's the part in the middle here, but we're looking at all 3 circles, not just the part in the middle here. Right? We're trying to deal with all 3 circles here. There's a talent, boy, 19 faculty recruited in the last 5 years here. Now, I know some have already left. Some have left and already come back. That's just the nature of academia. I get it. So we're going to take a pause for the next year or 2 until our endowment funds come back up from the Baker family. But we'll be going nonstop again, trying to recruit faculty because we've got to build up our talent here at UCSF. In terms of training, we've gone very horizontal. We're trying to get more people to learn how to write code. In fact, what we've been doing is partnering with the UCSF library to try to get more of those courses out there. Of course, we have great graduate programs. If you want a PhD in this stuff, of course, we got a PhD program. But instead, we want to make sure this is your chance to learn how to write code. Because you know what? You got 3rd graders in the state of California all learning how to write code. about time all of our students and maybe even faculty learn how to write some code. So really basic courses in R and Python, right? Very simple programming languages. I'm really proud to announce 1,400 new coders since we started this effort 5 years ago with the libraries. Incredible. But of course, if you want to go deeper, we got machine learning workshops, deep learning workshops, natural language processing, clinical informatics. That whole session's been going on with nearly 20 different seminars, all web recorded. So if you got a new fellow, if you got a new resident who's on a research block, wants to learn how to use clinical data, all of these courses are there, already pre-recorded, data.ucsf.edu. I'll be talking about that in a moment for you to get started. And we're constantly trying to figure out newer courses as well here. But in the last few minutes of time, what I'm going to do is really talk about the other big data. And that's really the data that got me out of Stanford 5 years ago to move here to UCSF. And that's electronic health record data. Yes, I know many of you are clinicians and this system really is painful, right? Epic. Maybe it's being less painful now that you've gotten used to it. But boy, are these systems expensive. First of all, it's expensive because we're paying doctors to type in all this data, right? I call this the most expensive data in America because of that. Because we're literally paying doctors to type in these notes, or at least click and point and click and all. But it's also expensive to put these systems into place, right? Look at these 3 press releases here. Sutter, right, our neighbors here in the peninsula and in California, spent $1 billion on their electronic health record system. Partners, which is Mass General and Brigham over in Boston, started with $1.2 billion for their electronic health record system. And Kaiser Permanente, spent $4 billion on their EHR system. And you want to know what's really amazing? They all bought the same system, Epic. That's what we use, right? I don't even want to count how much we've spent on this. But can you imagine the price tag can range from $1 billion to $4 billion? So the narrative I want to make sure you get, though, is if this is the most expensive data in America, and we've spent hundreds of billions of dollars on these systems, it will be a national tragedy if we don't use this data to improve the practice of medicine, right? Hundreds of billions of dollars later, and we still haven't improved what we do. That's a national tragedy. Of course, it's got to be safe, respectful use of data. I'm going to keep saying it again and again. Safe and respectful use. You got to follow the rules. But boy, if we don't do something with this data, what a tragedy it will be. And that's what I'm really talking about here. So with that, let me talk about the big picture here, right? Let's reintroduce the University of California to you, right? Of course we know UCSF. Some of you might have gone to other campuses for your undergrad or med school. We got 10 campuses, 3 national labs, 200,000 employees, right? We're one of the larger employers in the United States, and 250,000 students per year. 20 health professional schools now, right? We got 6 medical schools, but we also have nursing, pharmacy, dental, public health schools, veterinary schools, optometry school, right? So we've got 20 total. We train half the medical students and residents in California. So in other words, if they don't stay with us in UC, we're probably still having some influence if they go to Kaiser or elsewhere. $2 billion in NIH funding. We make $13 billion a year seeing patients. 5,000 doctors get a paycheck from us every month. About 100,000 doctors have written orders on our patients in the last 8 years. And of course, we're not random centers. UCSF and UCLA are in the top 10. Of course, we only mention that when we're in the top 10. Otherwise, we don't care so much for that metric. We have 5 NCI comprehensive cancer centers, 5 clinical translation science awards. And then we got these secret superpowers that are so secret, even we forget we have them, right? IRB reliance. If one of our med schools approves the IRB, the others automatically approve it, more or less, right? We can scale like crazy. Common contracting. If they sign it over in Oakland at the office of the president, all 10 campuses agree. They don't renegotiate. And what we're talking now is the umbrella across the University of California health system called UC Health. And what we've now built is the single central data warehouse for all of that patient data in one place. Of course, it's going to keep living on the campuses as well, but every month, and now with COVID, every day comes centrally to the University of California health system. That's the team that I'm privileged to run centrally to do this. Here's some of the recent numbers here. This is a busy slide, but we're talking about UCSF, UCLA, Irvine, Davis, San Diego, Riverside. Of course, Riverside is tiny. They're a brand new medical school. The other 5 are enormous. The top line number we all love to start with, whether it's right or not, is we've treated 15 million patients in the last 15 years. Now, it's a nice round number because that's 5% of the entire United States population. We love that number. But if you count from when we put EPIC in, right, January of 2012 at UCLA, UCSF, we've seen just short of 6 million patients since then, since January of 2012. 6 million, so that's 250 million encounters, 100 million procedures. My favorite, 600 million medication orders, 640 million diagnosis codes, 2.3 billion blood tests and vital signs. I like to say it's everything from Tylenol to CAR T cells, right? That's probably the least expensive thing we do is give a tablet of Tylenol, to the most expensive thing we do is to take your T cells, program them to fight your own cancer and shove them back in your body, right? So we got everything in between there, right? As well as those extremes. We got OSHPD data from the state regulatory agencies, pathology, radiology, text data, the death index, claims data, and all of those things here too. So I'm not going to go into details here, but this is what's available. Oh, by the way, 1 million of the data, 1 million of the patients are kids, okay? So you got 1 million kids in this database here, okay? Of the 6 million, 1 million of them are kids. So you got multi-center. Everything you do here is multi-center on arrival here, right? Because you got multiple, you got Mattel Children's. We don't have Rady Children's or Children's Hospital Orange County yet, but of course we got Benny Huff on both sides, Mattel Children's, and all the pediatric services across the UC. We've been using a lot of this to really help our operational teams. First 2 to 3 years of this has been decreasing our expenses on a self-funded plan. That's UC Care in blue and gold. Improving quality of care, especially for Medi-Cal patients, managing our costs, and then central management for primary care patients. I'll show you a couple of these in examples here. But here's an example of UC working together. And I want you to think about what this would look like in pediatrics. What would this look like in pediatric specialties? Right, Janet Napolitano and Jonathan Call in 2017 said all the cancer centers are going to come together, right, for clinical trials. The bigger we can represent ourselves to be, the more we're going to get those life-saving drugs to our patients in clinical trials. That's really the only way we can get some of those drugs to our patients here. And as an example of this, here you go. We saw 160,000 cancer center patients in 2019. That's our best guess is that's triple to quadruple the #1 center in the United States. We probably all know who they are down in the South, right? So we know we are stronger when we act together. Any one of our centers is tiny compared to MD Anderson, Dana-Farber, Sloan Kettering. You put us all together, we're enormous. This is what I mean by getting and competing for trials for our patients. But it's not just the sheer numbers. Now we got Foundation Medicine, some of you know, for the later stage cancers in kids and adults. Of course, we run UCSF 500. That data is coming as well. And we're still missing UC San Diego here. They're busy trying to get students back on campus. But here's an example how we now will have a single dashboard with all the gene mutations of all cancers across the University of California. P53 happens to be the gene that changes the most across all cancers, followed by KRAS and all the rest. No surprises there. But it won't be too long now before, for example, a doc at UCSF might see a cancer genome mutation and say, you know, maybe last year at Irvine, they saw a patient with that mutation, right? The patient might even have a different organ system cancer, but the mutations might connect us across UC. And maybe we can learn which therapies might have been successful or not successful there. Here's another example of looking at all patients, all primary care patients. Now, these are adults here with type 2 diabetes, right? A nasty thing to have. Of course, we see more patients with type 2 diabetes than this, but these are ones attributable to us for primary care. We're the ones tweaking the meds. And you can see here, okay, 44,000 patients, or just shy of that. This data is updated monthly for all primary care folks and all the champions on each of the UC campuses. One dashboard for all 5. Most of them are seen by a primary care physician, but some are seen by an endocrinologist. We got the latest A1C there. How much has it changed? It's getting worse for a lot of folks. COVID, right? Not walking around outside, maybe not going out much. And automatically, okay, this is with automated tools, we can look at eye health, kidney health, and then we can compare that to IHA benchmarks to know who's better, who's worse, what magic have you figured on your campus to make patient care a little bit better or not. So these are just examples of operational things that we've started to do with all of our data elements. But I'll say that the research side of this is really where a lot of this growth is going to happen. And just to skip ahead a little bit, how is research access going to work? we want you to learn how to access this data. But you got to start with our own campus first. So you got to learn how to write these kind of queries. They're called SQL. That's a code that you use using OMOP. That's the data tables. Patients go here, drugs go here. And we have ways to learn. I'm going to show you the slide in a moment. And then once you get your queries working at UCSF, and when you're ready, we will help you scale that question UC-wide, right? So we don't have a lot UC-wide over at UCOP to teach you or to get all the debugging to work. So you get that to work at UCSF first, and then we help you scale with this enormous database for de-identified data. And you bring your tools, you bring your code, we'll help you get it running. You can't download any of the data, and you got to sign your firstborn child away if you do something nasty with the data, like download it or give it to a company. But that lets all this data be available to you for research questions. At UCSF, you would start with data.ucsf.edu. This is your front door to getting to clinical data. Is it for quality improvement initiative? Is it for research? All of that happens here first. So I'm going to stop there. And usually I would stop here and show you some glitzy animation. But because there are trainees in the audience, I want to spend one last minute just talking to trainees here. So of course I'm a pediatrician, right? Technically, I guess once you're a pediatrician, you're never not a pediatrician, right? And look, I'm not active clinically, or as the joke goes, I do still see patients, but I see them in the cafeteria in the hall here, right? I'm not seeing in the exam room here. So maybe my life lessons are useful or not. But here are my life lessons to you as trainees here. What did I learn along the way? We're only as junior as we want to be. What that means is I love and yet hate this phrase junior faculty, because somehow junior faculty seems to mean to some folks that you should ask small questions and kind of don't tip over the apple cart or whatever. I think junior faculty should be a state of funding, not a state of mind, okay? I love it when my mentees and my trainees kick my ass. I know my mentor, Zach Honey, loves it when I do it to him. Of course, you should keep challenging as far and as hard as you can. Okay, don't go in a timid way. The whole point is to try to go for changing the world in the questions you're asking. Set the level of your peers as high as you can. Yes, the folks you might be on call with might not be writing papers, but the ones at Boston Children's and everywhere else are writing papers. Look nationally, look internationally to figure out who are your real peers here. Train as much as you can afford. I went back after my fellowship to go get a PhD. Not everyone can afford to do that. I totally get it. But if you can delay that gratification even a little bit more, it doesn't hurt to get a master's degree, other training, maybe even a PhD, who knows. Don't be afraid to innovate beyond your university. Industry and startups are all on the table. Of course, there's rules and ways to do it. They're all achievable ways to do it. Solve big real world problems. Yes, impact factor and H-indexes are great. Of course, I follow mine too. I'm that vein, but they're just games here, right? Real impact comes from real reach to patients and to families and doctors. We in pediatrics are too small of a field, okay? Not only should every patient in everything be in a trial, we should be learning from every single patient, okay? And I know we get in cancer. I don't understand why we don't get it in everything in peds still. Communicate and cheer your colleagues to get more total funding into pediatrics. I don't understand why we do it, but we sometimes unnecessarily bring others down. That's crazy. We need more general funds to lift all the boats in peds. And you determine your future, not NIH. I still run into too many trainees that say, if I get funded with my K award, I will stick around in academia, okay? It's the wrong way to think, I think, okay? What you gotta start first is say, I will make it in academia and eventually NIH will find me. And why I say that is, this is my first pink sheet, okay, back when they were pink sheets, and these were my scores for my first K award application. And back then they ran from 100 to 500. 500 was the worst possible score you could get. And now back then they would have triaged, now they would have triaged this. They didn't triage them back then. 414. This is as bad as you get. It was probably the worst score in the study section. And I'm a survivor, okay? So you start with this, it only goes up from here, okay? So with that, I wanna thank an enormous number of collaborators who get all this work done. My friends at Stanford, many more friends now at UC and UCSF and at NIH. Incredible team that helps put all this clinical data across the campuses. A lot of support. I have to thank Carrie Byington a great pediatric infectious disease expert, and of course, the executive vice president for UC Health. I have to thank incredible funding from UCSF, Priscilla Chan, Mark Zuckerberg, the Baker Foundation for their naming gift. My lab has been blessed with 20 NIH grants from these 11 institutes of NIH. The ones in red give me more money. The ones in orange give me less money, but I still love them. All sorts of foundations now have given us funding to work on their diseases, maybe even come up with therapies. On the top right there, my admin and tech staff, I'd never get a grant or paper or project out the door without them. I thank many members of my lab over the past 20 years. I've named some of them on some of the slides. And I have to thank Zach Gunny, my friend and mentor for life, Sam Hagen, Keith Yamamoto for recruiting me, Talmadge, Mark, Jack, and now Carrie Byington for their support, and my wife and daughter who really inspire me to keep all this work going. So let me stop there and see if I can take some questions. Thank you.
Well, thank you, Atul. That was marvelous, fantastic. I'd like to maybe start with a question. You and I had spoken a while back, shortly after I got here, and you had mentioned how, even though you're in the Department of Pediatrics, most of your you don't have that many pediatric collaborators. And one of the reasons I wanted you to come to do Grand Rounds is so that people could be more aware of your program and the opportunity. And I just wonder if you could maybe take a moment to make some suggestions to the department about how they can better utilize the resources available through your program, and what kind of things you'd like to interact with folks about.
Yeah, absolutely. So first of all, it's not that I'm avoiding pediatrics. One of my postdocs and staff members who came over from Stanford, Marina Sirota, now is an assistant professor. I think she's now promoted to associate professor. Really has been doing a lot of that. And kind of to stay out of her way, I've let her take in a lot of the OB and pediatric collaboration. So I tend to do that with trainees. I give them their space so that I'm not stepping on their collaborations and stuff like that too. Now, that being said, I now have formally signed, I think, 1 or 2 agreements now. So I do have pediatric researchers who are trying to work on their care wards with me. And so in that mentor-mentee relationship probably works well. And, you know, I will never say no to that. So I think I'm there to help, especially on the quantitative side, computational side, for a lot of projects for some of the more junior folks who are looking for help and mentorship for their care wards. So I think that's probably the best way that I can relate with folks. Now, that being said, we have all this clinical data. So we got teams that are there ready to help. I would love for people to use the data at UCSF, get some queries asked and answered, find some science that you're interested in, and then we'll help you scale UC-wide when you're ready and we're ready.
Wonderful. Let's see if we have any questions. Sandrain, are you collecting questions? I can't tell from this.
Yeah, so there's currently actually one question on the chat, which actually says that the person would be willing to take it offline as well. But since we have a minute, the 6 university rules for spinning off a startup that you refer to. Can you elaborate?
Yeah, so basically, let me see if I can get them off the top of my head here. So the first is, first and most importantly is you got to disclose everything, okay? And ironically, in some weird world, the more you disclose, the more trustable you are, right? So it's kind of weird, right? And of course, I had to provide these slides ahead of time, right? But it kind of looks kind of suspicious when you have one company. The minute you have 100 companies, somehow it seems more believable that you're trustable. I don't know that irony. Disclose everything is the first rule. The second is, if you're doing research projects, you cannot have your company doing clinical trials with your patients, right? That's a big no-no. You got to separate those, right? Your company's got to go do things with other people's patients. Another rule is your students can't be in the company with you. Why is that? Because they have to have freedom to publish. So your company might inadvertently tell them not to publish something, but they need publications to graduate with a PhD. So there's even instances where my students have joined companies, but I have not until they're done, and then then I'll join the company. You can't pipeline. In other words, everything you keep inventing in your company has still got to go through, I think we call IPA here, industrial partnerships, right? You got to disclose everything. Another rule is your company usually cannot fund research back in your lab. You've got to kind of disconnect there. And in general, what that leads to is it's really tough for your lab. It's tough when your lab competes with your own company. So usually when it gotten enough to start a company, my lab shifts slightly in a new direction. Because it's really tough to compete with your own company. It's not rewarding at all. So I think that was a 6 bullets. I'm probably missing one there. But in general, those are the same rules everywhere. We got to disclose everything. Oh yeah, there's time and hour restrictions here too. So usually most universities let you have a day a week. I think for us, it's like 384 hours. So I count it by the hour. But however you count it, that's how much time you're allowed. It's 20% time. But you got to count every hour. Okay, don't approximate. Don't do any of that. And then at UCSF, we have a $1 threshold too. It doesn't mean you can't make more than that, but you got to cut off a fraction of that and give it to the university above that $1 threshold amount. And all of that is in the comp plan.
Great, thank you so much, Atul. This was fantastic. It's 1:02, so we should probably let people go. Dr. Hirsch, what do you think?
Yes, I agree, Sandrain, and I'd like to thank Atul one more time for a fantastic presentation, very thought-provoking and stimulating, and wish everybody a good rest of your day.
Thank you, and stay safe.